We're now going to talk about the different types of vaccines. And for step 1 USMLE, this is going to be really high yield. It integrates obviously some microbiology and some immunology. And for each sort of infection that we do have a vaccine for, it's going to be important that you know what type of vaccine it is. And we're going to start with live vaccines. And then you really need to know about the type of immune response that each vaccine elicits. So we're going to start with live vaccines, and for the most part, of course, live vaccines are attenuated. And when you hear that word attenuated, essentially that just means that it is weak. So it is incapable of causing disease, hopefully, in the host. So we take live pathogens, and there are several different ways that you can attenuate them. I'll give you an example of the vaccine for TB, which is a strain of Mycobacterium bovis, was grown for a period of over 10 years in bile, which caused it to become weak and incapable of causing disease. So there's lots of different crazy things we do like that to cause something to lose its capacity for virulence. When an organism doesn't need to be virulent anymore, under different circumstances, of course, it can lose those properties. The best thing, though, about these attenuated live vaccines is they still replicate in the host. And that's going to be important for what we're going to talk about in the next slide. These vaccines are really the best vaccines because they essentially mimic the natural disease. So they give us both a humoral and a cell-mediated immune response, including the B cells and T cells in that response. And because they mimic the natural infection, we typically get lifelong immunity. Now, one dose usually provides immunity, and what that means is, so let's just use MMR as an example. We give MMR typically after 12 months, so between 12 to 15 months of age. That usually provides immunity, but not in everyone. So we always give a booster dose, usually right before the kids enter into elementary school or kindergarten, and that usually ensures that they seroconvert, meaning they become immune. And that happens, again, that helps most individuals become immune. Now, even though these live vaccines are the best vaccines at giving us immunity, they are dangerous for certain individuals, and we consider them dangerous really for immune-compromised patients. So in these individuals, even viruses that are attenuated can cause disease, and therefore, on step 1, which does vary a little bit from reality, and I'll discuss that in a minute, on step 1, we are not going to give a live attenuated virus to anyone that's immune compromised. And why I say that basically veers from reality a little bit is, let's just use an AIDS patient, for example, okay? They're going to give you some scenarios of AIDS patients and, what vaccines are safe. And of course, you're not going to pick one that's live attenuated, okay? And on step 1, it's sort of a yes or no answer. But when we get into step 2 and step 3, it's going to be important that you actually look at the T cell count. So obviously, if you have an HIV positive individual who has a good T cell count, we're going to want to vaccinate them to protect them for when they are in full-blown AIDS. Another patient population that falls under this category are pregnant women. We don't often, maybe think of them as immune compromised, but we have to try to remember that in pregnancy, really a woman's body is trying not to reject the fetus. So instead of being super immune, like we might think she should be, she's actually a weakened immune system to try not to reject the fetus. Attenuated vaccines, we said, are live organisms. They're just weaker and incapable of causing disease. But with any live organism, there's always the slight chance or potential for it to revert back to a virulent form. And this is a really important point, and it's the precise reason why in the U.S. we no longer use the live polio vaccine. Of course, the Sabin vaccine is the best vaccine. We get really good IgA immunity. And we would actually shed that vaccine in the feces for about a month, just like we would live polio. Okay? But because polio has been eradicated from the U.S., we now no longer give that live vaccine because you would have a better chance of getting polio if we gave you the live vaccine with the slight potential for it to convert back to a virulent form than if, of course, we gave you the killed vaccine, which is what we exclusively use in the U.S. So the following live vaccines are recommended in the U.S., which of course means any one of these that are recommended in the U.S. are high yield and there's a good potential for them to be tested on step 1. So obviously we've already mentioned the MMR vaccine. The VZV vaccine is actually similar in composition, whether we give it to prevent chickenpox in young children or zoster in the elderly. The rotavirus vaccine and the flu mist. So this is the live attenuated version of the influenza vaccine. Now, we do have some other live viral vaccines that you could potentially get in the U.S., but they're only recommended under certain circumstances. So we mentioned the Sabin vaccine. This one, of course, we don't use anymore because it would potentially increase the chance that you would get polio. But, of course, if you were traveling to an area where polio was endemic, this is the best vaccine to give. So there are certain circumstances where we may give that. The smallpox vaccine is, of course, only given to military personnel. And, of course, that's a really unique situation because this virus has actually been eradicated from the entire globe. And then, of course, the yellow fever vaccine. And you always want to remember about that yellow fever vaccine because they can ask nice little sneaky questions about the yellow fever vaccine. For example, let's just say they gave you a nice little vignette about a patient with yellow fever and then asked you what would have been the best mechanism of protection to prevent the illness in that patient. And there's two answers that are going to drive you crazy. One of them you should always pick, which is vaccine, because a vaccine is always going to give the best mechanism of protection. But another potential thing they can put in the distractors is mosquito netting. And I've seen you guys do it a million times. And what you do is vaccine's the right answer. And then you're going to look and you'll be like, oh, yellow fever is transmitted by mosquitoes. And yeah, but don't do that, okay? Because mosquito net never as good as a vaccine. And of course, a lot of these mosquitoes that transmit some of these viruses are day feeders. So then what you're going to do? Walk around with a big old mosquito net all day long. Nope. So don't pick that one. Now, there is one really unique example of a live vaccine that is actually non-attenuated. Yep, you heard me right. A live vaccine that is fully virulent. And of course, if anybody's going to use that vaccine, you can bet it's going to be the U.S. military because they're tough, right? So they use this sort of vaccine against adenovirus serotypes that are very common in causing an adenovirus pneumonia or acute respiratory disease in military. So it's a very common thing for recruits when they're in basic training to get adenoviral pneumonia. So they developed this vaccine that is given in an enteric-coated capsule, okay? And this capsule then releases the virus in the gut when it dissolves and produces essentially an asymptomatic infection that gives us really good IgA response. And the most important thing that you need to know about this, of course, is that IgA is not just protective in the gut, but it's protective at all mucosal surfaces. So that IgA then, even though that response was generated in the gut, it will protect in the mucosa everywhere. So the respiratory tract will be protected, the genitourinary tract will be protected. And the reason this works, if you go back to your microbiology, is because, of course, This is a non-enveloped naked virus, so it can withstand being in the gut. So these vaccine recipients are protected against adenovirus acquired anyway. It doesn't even have to be aerosol, but that's sort of what we're trying to protect them against in this special case. And it really is the only example of a live non-attenuated vaccine. So occasionally we know that USMLE likes to test on the zebras instead of the horse. Another vaccine type would be the killed vaccines. And the killed vaccines essentially just use whole organisms that are killed, usually not by heat. They can no longer replicate, so in general they're very safe. And as I just mentioned, they're inactivated usually by chemicals rather than heat, because if you think about what happens, of course, when you fry an egg, you're denaturing those proteins, and actually you are destroying the immunologic epitope. The difference, if you're comparing live and killed vaccines on a few concepts, of course, these require several doses to give you the desired response. And remember what we said, most people after receiving a live vaccine will actually convert to becoming immune. Now, with killed vaccines, these usually just give you a good humoral immune response, which is perfect, because if we have a good high antibody titer, then we can prevent these infections from even initiating. The killed vaccines that we use in the U.S. are rabies vaccine, which is a unique vaccine in and of itself, of course, because we only use that post-exposure. So most of these other vaccines really were giving to prevent disease at all, but rabies vaccine is really supposed to be given after bite of a suspected rabid animal. So that's a little bit unique. The influenza vaccine, the injectable vaccine, of course, is a killed vaccine. The vaccine that we predominantly use in the U.S. for polio, which is the Salk vaccine, and then the Hep A vaccine. And if you look, you can remember these by looking at some of the letters here. Rest in peace, so RIP always, the A from hepatitis A, and that helps you remember that those are killed. And again, those are generally pretty safe. We can give them during pregnancy, and they provide a good enough immunity to help protect us against these various pathogens. Toxoid vaccines are sort of another unique type of vaccine. These guys are actually made from inactivated exotoxins from bacteria that secrete those toxins. And the reason that we said that they're somewhat unique is because all other vaccines that we have are there to prevent the infection itself. But toxoid vaccines specifically prevent the disease, but not necessarily the infection. So let me talk about the different toxoid vaccines, and then I'll give you the examples, okay? So diphtheria, so that's Corynebacterium diphtheriae, Clostridium tetani, and then Bordetella pertussis. All right, this is the DTP or the DTaP vaccine. So because these vaccines are aimed against the toxin, so these are inactivated toxins, it's not going to prevent you from getting colonized with Corynebacterium diphtheriae, but what it will do is prevent you from getting the disease because you have antibodies against the toxin, and these diseases are mediated predominantly by the toxin. So when the bacteria begin to secrete the toxin, your antibodies will bind the toxin and prevent the toxin from binding to the host receptor. So again, unique situation where we're preventing the disease itself, but not the infection. So you may have noticed that sometimes you'll see the DTaP vaccine and then sometimes you'll see the DTP. When you see DTaP, that means that the vaccine is completely acellular. So that's what the A actually stands for in front of the P, acellular. And it's composed of toxoids, or even for pertussis, some filamentous hemagglutinin from those organisms. Now, in the DTP vaccine, the pertussis is actually whole killed cells. And so that's going to be the difference between the DTP vaccine and the DTaP vaccine. The DTaP vaccine is considered a little bit safer with fewer side effects, and we currently pretty much exclusively use the DTaP vaccine in the US. So you'll want to pick that one. Polysaccharide vaccines, we used to have actually quite a few of these. We now only have one major one that we use, but these guys are comprised of really just polysaccharide capsular material that's found on, of course, the encapsulated bacteria. But if you go back to some of the basics that we talked about earlier in this book, we know that, of course, polysaccharides cannot activate T cells. So without the help of T cells, these guys can only produce IgM, right? So polysaccharide are not going to go through that T helper cell pathway. We're not going to get any class switching. So we're just going to get a IgM response. For the most part, these vaccines have been replaced with conjugate vaccines, which I'm going to talk about in a little bit. And those conjugate vaccines are a lot better because they can induce class switching. When we're talking about these polysaccharide capsular vaccines, we said they're only capable of producing IgM, but another sort of important feature about them is we're not capable of getting a booster response, because without being a T cell dependent sort of antigen, we're not creating immunologic memory here. So we only get IgM, and we never really get a booster response when the T cells are not involved. So the current only polysaccharide vaccine that we use right now in the US is the adult pneumococcal vaccine, or the PPSV23. This vaccine is comprised of 23 of the common capsular serotypes associated with the most severe disease in that age group. So typically, it is indicated for adults that are 65 or older, or, of course, individuals that have special circumstances, whether it's COPD or, of course, splenectomy. Splenectomy is going to make you susceptible to all of the encapsulated pathogens, and that, my friends, is a question that will plague you for the rest of your life there. When you get out on the surgical wards, step 2s, step 3s, they're always going to ask you what sorts of things you want to vaccinate for and why, if you are about to do a splenectomy. So another really important vaccine type, especially for step 1, are the conjugate vaccines. For the reason that we just mentioned with the polysaccharide vaccines, which is that they can only induce an IgM response. So what they did was they conjugated this polysaccharide to a protein, which created this way better immune response because, of course, it made it T cell dependent. So with these conjugate vaccines, which again are made up of the capsular polysaccharide from the bacteria that has been conjugated to a protein, we get class switching, we get a memory response, and because we get a memory response, we actually get a nice booster response to those multiple doses. The first vaccine that actually made the transition from a sheer polysaccharide vaccine to a conjugate vaccine was the Hib vaccine. And in the next slide, I'm going to show you essentially how those work. But that was the first one. But currently, of course, we have a few more of these conjugate vaccines. We have the pediatric pneumococcal vaccine, which is comprised of 13 different capsular serotypes, again, most often associated with invasion and indicated for use in infants. It's part of our recommended vaccination schedule in the US. Neisseria meningitidis is the other one that is now conjugate. So again, these all used to be capsular polysaccharide vaccines, which were good. They provided immunity. They gave us some IgM. But again, by conjugating them to protein, we allow the T cells to get involved and we get a much more vigorous response. So let's take a look at how a conjugate vaccine works. So I'm just going to give you the lay of the land here. On the top, we're looking at our T cell. The bottom cell here is the B cell, and you can see this antibody in the membrane. That is the B cell receptor. Okay. Now this big pink and blue thing is actually the vaccine. And the pink stuff that looks like sort of that pink slime that they're talking about that a certain fast food chain may use in their beef and whatnot, that pink slime is the carrier, which is the protein or the peptide, okay? But the epitopes that are being recognized by the B cell receptor, that comes from the capsular polysaccharide. And here they're calling it CHO, which stands for carbohydrate. They're also calling it a hapten because, at least in this example, they're really small and would be ignored by the immune system, except for now they're linked to a huge carrier protein, okay? But because they're linked to this huge carrier protein, they can get processed and put onto an MHC molecule. Remember the exogenous pathway for antigen presentation that we talked about? So that's what's happening here. This antigen gets processed, and now we can present protein to a T cell, and that T cell then becomes activated and will now begin to give this B cell some cytokines to help it activate and proliferate and undergo class switching. So what that B cell is now going to be able to do is turn into a plasma cell that secretes really nice, high affinity, hopefully antibodies against the capsular polysaccharide, which is actually the major antigen in this situation. So these conjugate vaccines, I'm telling you, Step 1 USMLE loves questions on the conjugate vaccines. So you really have to know which vaccines are conjugate vaccines and what's the big deal about them. And of course, the big deal is that now they're T cell dependent by conjugating that capsular polysaccharide to protein. So we're getting class switching, and we're really getting the benefits of a booster dose because we're getting a memory response. Alrighty, now the last vaccine type we're going to talk about are the component vaccines. And component vaccines are actually really good vaccines because instead of, let's say, killing a virus and using all of that virus to create a vaccine, well, we don't really know all of the different proteins and such that the immune response in that case is going to make an antibody against. But here, we're taking the most immunodominant protein, so the one that we know gets a robust response from the immune system, and we're utilizing that as the vaccine. So that gene for that protein is actually grown in yeast cells. And a really nice example of this is, of course, the hepatitis B vaccine, where we take the gene that encodes for the surface antigen and insert it into yeast cells. And the yeast releases that molecule into the culture media, and then we purify that and use that as a vaccine. Now, why this is such a really good idea is because, of course, we know that if we get antibodies to the surface antigen in the case of an immune response to hepatitis B, that provides immunity. So we don't need to worry about antibodies to the core and other antibodies that might be formed if we used a killed vaccine. We're just focusing on the one protein that we know if we get an antibody response to will give us immunity. So as we mentioned, hepatitis B vaccine is comprised of the surface antigen, but another good example of a component vaccine is the human papillomavirus vaccine. The original vaccine was a quadrivalent vaccine made up of 4 different serotypes, 6, 11, 16, and 18, and you guys should know this from the microbiology. Serotypes 6 and 11 were the serotypes that caused over 90% of genital warts. So those are the serotypes associated with genital warts. And then 16 and 18 were the most likely serotypes to cause cancer, and therefore would protect up to about 70% of cancers. There has been a new vaccine now, the Gardasil 9 vaccine, and essentially it keeps 6 and 11, it keeps 16 and 18, but includes now several other serotypes so that we can prevent over 90% of cancers due to HPV. This one's relatively new. This one was released in just February of 2015. So anybody taking a recent version, hopefully, of step 1 for USMLE, this may or may not be on there because we know there's sort of a lag period from the time that we get new vaccines or we see new pathogens like Zika. Is going to take a little while for that stuff to make it onto the step 1 exam.